This is the 2022 third quarter online investor conference of Oneness Biotech Company Limited. We are very pleased to be joined online by the distinguished guests and the investors in this virtual investor conference. First of all, I would like to introduce to you the management team: General Manager Qihui Cheng, Vice President of Medical Science Shai-Jen Chen, Vice President of the Antibody Development Nian Chen, Vice President of Small Molecule Development Nina Wang, Senior Manager of the Finance Department Amy Siyao. Next, I would like to invite Qihui Cheng, the General Manager, to give a brief speech.
Shareholders, ladies and gentlemen, good afternoon. It is the 2022 third quarter virtual investor conference of Oneness Biotech. The management team will report to the shareholders on the progress of the research and development of the new drugs and the explanation of important subjects the shareholders mostly concerned. I hope to let shareholders know more about Oneness. In the third quarter of this year, under the continuous epidemic situation, the clinical trials of two important asthma new drugs, FB825 and FB704A, have indeed been affected. We slowed down the patient enrollment progress of the trial in Division of Thoracic Medicine to minimize the impact by the epidemic situation on the trial quality. However, the important research and development results of Oneness are still quite fruitful. Oneness medical device for wound care has finally obtained the FDA 510(k) clearance supported by the regulatory science. This represents the world's first-in-class wound care cream of Oneness achieved the strategic goal of the U.S. market entry under a dual product strategy with different new drug and medical device. This is important for global market access of Oneness integrated wound care products. Secondly, in the face of the ever-changing coronavirus, SNS-812, a nucleic acid new drug co-developed by Oneness and Microbio Shanghai, was completed with the drug GMP process, quality management and preclinical toxicology studies within 11 months. It has been successfully greenlighted by the FDA to proceed with the U.S. Phase 1 clinical trial this month. It is currently the only new drug in the world that can inhibit different variants of coronavirus. Meanwhile, 99% of the infection prevention has been confirmed in vivo. According to the latest international research, there are multiple variants of Omicron, including BA1, BA2, BA5, BA2.75.2, BQ1.1, and so on. Among them, BA2.75.2 not only results in breakthrough infection, but also escapes the human immune system, and it is expected that this winter will be a cold winter where viruses are raging. We have expedited all possible procedures, with the goal of completing Phase 1 trials by early next year and entering Phase 2, hoping to accelerate the efficacy confirmation of SNS-812 in viral suppression in humans to get marketing authorization as soon as possible. The NDA of ON101 in China that everyone cares about has now entered the final critical stage, and the required documents and clarification have been fully submitted by Oneness. From the regulatory and scientific perspectives, the requirements should be satisfied. And now we must wait for the decision by the health authority. Considering patients' standpoints of urgent need, we look forward to ON101's approval in China as soon as possible in accordance with the regulations. As for the international development, the U.S. Phase 3 trial is still in progress, and we have also taken the opportunity to discuss with the FDA on the regulatory requirements for the NDA filing in the U.S. The regulatory communications are still ongoing. We are also working towards the future licensing deal opportunity. The momentum of innovation and R&D of Oneness continues to grow, and the number of drug pipeline has now increased to 6, including 5 new drugs and 1 medical device, each of which comes from breakthrough innovation and research and development. Although there will inevitably be uncertainties and challenges in the process, Oneness team would be capable of handling all the challenges of new drug development on the basis of diligence and professionalism as well as of science, integrity and transparency. And I believe that Oneness will continue to create a greater success. Thank you. This is the disclaimer of this legal lecture. Please refer to it.
Next, the research and development progress will be reported by Shai-Jen Chen, Vice President of Medical Science.
The research and development progress report of Oneness new drug includes 6 major pipeline Firstly, FB825 is an anti-SAM-X antibody new drug. The report will include subsequent planning for atopic dermatitis and a phase 2 trial of allergic asthma. Secondly, the progress of FB704A, an anti-IL-6 antibody drug, includes the execution of phase 2 trials and the development of a second indication. The third is ON101, a breakthrough macrophage-regulating new drug, including the U.S. Phase 3 trial, regulatory update in China and other Asian countries on NDA progress, international publication, and medical industry academia research collaboration. The fourth is topical medical device, including 510K in the U.S. and MDR in Europe. The fifth is OB318, an anti-cancer drug derived from Entrodia campharata extracts that is currently under the Phase 1 clinical trial. The sixth is SNS812, an anti-coronavirus siRNA drug, and is currently undergoing a Phase 1 clinical trial. For the antibody drug, we invite Dr. Nianyi Chen, Vice President of the Antibody Development, to report the progress.
Thank you. Then I will be reporting to you the current progress of the development of the antibody new drugs. First of all, I will report the planning of FB825 studies for moderate to severe atopic dermatitis. In June this year, we have completed the clinical study report of the Phase 2A clinical trial in the US and have also delivered it to our partner. Oneness also continued to move this product forward. In September of this year, we have submitted a consultation meeting to the FDA on human pharmacokinetic bridging trial of subcutaneous injection and intravenous injection as well as the Phase 2B clinical trial protocols. This meeting has been granted by the FDA. We will continue to follow FDA's advice on the future trials accordingly. This bridging trial is tentatively scheduled for Q1 of 2023. In addition, in the drug preparation, we have completed the production plant schedule for the subcutaneous dosage form study drugs, and the completion of production of the study drugs is tentatively scheduled for Q1 of 2023. The progress of another Taiwan Phase 2 clinical trial of FB825 for the moderate to severe allergic asthma has indeed been affected by COVID-19 in the past, and the progress of patient enrollment has slowed down slightly, but we have now 6 medical centers activated for enrollment, and also added NTU Hospital to our clinical sites. In addition, we are actively under contracting with more hospitals with an aim to increase the number of hospitals to 15, which can accelerate our clinical trial process. It is expected that by Q4 this year, these 15 hospitals will be activated to accelerate the progress of our enrollment. With the pandemic expected to ease, our trial schedule is planned as follows. The trial is scheduled to complete the enrollment in first half of 2023. The asthma trial duration is 9 months, so it is expected that we can complete the CSR in 2024. The above is the progress of FB825 in moderate to severe allergic asthma. Basic research and patents are important to a new drug. With regards to the basic research of FB825, we also actively prepare a manuscript for publication on valuable research findings. We are also actively planning patent portfolio. According to the results of FB825 atopic dermatitis exploratory experiment, the mechanism of action of FB825 and the target cells, we have noted a very strong correlation between the target cells with atopic dermatitis. As a result of the trial, we are evaluating its patent applicability, and we will continue to extend the global patent protection of FB825 with a wide range of patent portfolio. In response to the results of 3 exploratory efficacy trials that have been completed, we have also written a manuscript for submission to an international journal in Q4 to increase the international visibility of FB825. The target cells identified in this paper also involve the portfolio of our new patents in the future. In this exploratory experiment, we got results and we defined a very important subset of key cells to atopic dermatitis. This group of cells is the target of FB825. Of course, we are also evaluating the feasibility of patent protection. This chart reports to you where the newly discovered cell subset targeted by FB825 is distributed and what its importance is. On the left-hand side of this picture, it is the allergen from the outside into the human body, recognized by our immune system, and then these allergens are presented to our immune system, so that it can carry out a series of processes such as IgE production. Of course, the well-known mechanism of action of drugs in the past was related to cytokines such as IL-4, IL-5 or IL-13, so that the B cells are activated and differentiated into IgE. This type of B cells will release a large amount of IgE, causing the release of histamine. In the tissue of the skin or of the pleural cavity or of the trachea, its mast cells will release histamine. We will therefore experience the red itching of the skin, the asthma of the trachea. This is the result of this signaling pathway. This pathway will gradually deteriorate because the tissue released by this mast cell will further return to our immune system to stimulate such a response, creating a vicious circle. This is also an important mechanism for allergic diseases such as atopic dermatitis and allergic asthma. In this mechanism, we can see that the IgE B cells shown in this red box in the middle are the target of FB825. As a result of our new exploratory experiments, we have defined the subset of very important cells, so we will carry our further studies on these cells in the future. On the one hand, this is associated with the patent protection on the mechanism of action of FB825 itself. It is also possible that from this mechanism, we can explore new treatment strategies, so this is a very significant progress in our research and development. That's the FB825 report. Next, I will report to you on the progress of the Phase 2A clinical trial of FB704A in severe asthma. In this trial, we currently have 5 medical centers in the process of enrollment, and we have also kicked off the second stage of the site's evaluation. We expect to increase the total number of hospitals to 8 by the end of the year, to accelerate the enrollment. Of course, in the view of the epidemic situation to be eased, the enrollment of Phase 2A clinical trial is scheduled to be completed in the first half of 2023. The entire trial duration is 4 months, so in the second half of 2023, we can complete this trial. In FB704A, there is a very important biomarker study in treatment of severe asthma, which helps us to better understand the disease as well as the patients. In this exploratory biomarker study, we collect the sputum of patients for analysis to explore the respondents to this drug. This part of the collection is scheduled to be completed in Q4 of 2022. Since FB704A is the world's first biologic for severe asthma with high neutrophils, through this biomarker study, we can have a clearer picture of the patient population. It is possible to expand the target patient population and can further strengthen patent protection of FB704A. FB704A as an anti-IL-6 antibody, of course, its applicable indications are very extensive. We are also actively expanding its development to the second indication. The second indication is chronic kidney disease complicated by cardiovascular disease. As reported last time, the results of our in vivo and in vitro studies were completed. We are now evaluating the results of these studies to support the use of FB704A in this indication. Based on our comprehensive assessment, we concluded that FB704A can effectively inhibit key indicators of chronic kidney disease, such as haptoglobin and neutrophils. With that, FB704A has the potential to be developed to treat chronic kidney disease. Up to 700 million people worldwide are suffering from the chronic kidney disease. 50% of them will have complications of cardiovascular disease. There are not many competing drugs in this market at present, so it can accommodate multiple biologics, and FB704 still has a good opportunity in this market. We are currently planning to conduct an exploratory study to evaluate the efficacy of FB704A. IND submission for this study is scheduled to be filed in Q4 this year. This is the progress report of the antibody new drugs. Next, I will invite the Vice President, Dr. Shijin Chen, to continue with the following report on ON101.
ON101 Phase 3 clinical trial in the U.S., which is a pivotal trial related for marketing approval in the U.S. We have been in communication and consultation with the U.S. FDA to confirm the requirements on subsequent NDA. The execution of this Phase 3 trial in the U.S. will follow the instructions/recommendation by the FDA. Oneness has also completed the submission of the waiver request on pediatric study plan and the carcinogenicity studies. This trial is affected by the pandemic, but with the FDA's recommendations for the NDA, we are increasing the number of the clinical sites to meet the goal of filing the NDA in 2025. For the NDA progress in China, we have completed the response to the information request by China CDE on August 26, 2022. At present, the National Institutes for Food and Drug Control in Beijing is performing inspection on the delivered samples. The testing results will be submitted to CDE by the National Institutes for Food and Drug Control. As mentioned by our general manager, it is now a critical stage of NDA in China, and the subsequent progress will be subject to the review timeline by the health authority. With regards to Fespixin's entry into Asian market, the countries planned in the first stage include Singapore, Malaysia, and Thailand. Oneness has completed the response to information request by the Singapore HSA in August 2022, and also completed the response to information request and sample inspection by the Malaysian NPRA. We are under contract signing with an agent for Thailand submission now. We expect to submit the NDA application in the fourth quarter of this year. The second stage of Asian market entry include India, Vietnam, the Philippines, Indonesia. For India, the clinical trial waiver communication meeting was held by CDSCO in August, and additional data will be presented in accordance with the opinions of the health authority. For Vietnam, the DAV has accepted the NDA in July this year, and the review period is 20 to 28 months. With regards to Philippines, the FDA in the Philippines also accepted our NDA application in April this year, and the review period is 254 days. At present, we have not received information request by the health authorities in Vietnam and the Philippines. In Indonesia, the submission of samples has been completed in September this year, and an administrative review of the NDA is scheduled to be filed in the fourth quarter. For the third stage, we plan to submit a waiver request of clinical trial in South Korea. This is scheduled for a submission in October this year. An administrative review of the NDA will be submitted upon waiver grant. Next is the progress of medical device in the United States. The name of the medical device in the U.S. is Bonvadis, which is a cream for external wound care. On August 20th of this year, the U.S. FDA pre-market notification 510(k) for substantial equivalence and permission to be marketed was received. This product can be marketed after obtaining an export license in the first quarter of next year. The next phase of planning is based on the abbreviated registration with U.S. FDA 510(k) in other countries. Our consideration includes the quicker entry, and in Stage 1, we will include India, South Africa, New Zealand, and Australia. As for the progress of medical materials in Europe, the quality management system of scar management medical device have been ISO 13485 certified. The application for the European Union's MDR, which is the regulatory specification for medical devices, has been scheduled for the fourth quarter of this year. The next stage of planning for topical wound medical device. In terms of quality system, it is scheduled to complete the addition of medical device for wounds in the first half of 2023. In terms of MDR, applications for wound care medical device are scheduled to be submitted in the second half of 2023. With regards to the patient recruitment of the Phase 1 clinical trial of OB318, a new anti-cancer drug, we have successfully completed the second cohort group. During this season, we also successfully completed the third dose group, and the SRC meeting is expected to be held in October. After this meeting, we can start the clinical trial of the fourth dose group. And what is the difference between the fourth dose group and the previous ones? The previous dose groups must complete one patient before recruiting another one, while the fourth dose group can recruit 3 patients at the same time. It is expected that we can accelerate the trial by completing patient recruitment for the fourth and the fifth dose groups in the fourth quarter. In terms of the research and development progress of SNS-812 nucleic acid new crown drug, according to our latest database search on September 14, 2022, SNS-812's coverage rate for COVID-19 variant strains remains at 99.8%. New variants of Omicron such as BA2, BA4, BA5, and etc. are still within SNS-812's effective inhibition range. We have completed the synthesis, filling, and bottling of cGMP for SNS-812, and the drug's purity and data have met the standards. Toxicological tests under GLP were also completed, and no apparent drug-related toxicity was observed. We have filed an IND application to the FDA in the United States on August 23 this year, and on September 23, we have received a green light to proceed with the Phase 1 clinical trial. The subsequent plan for SNS-812 in treatment of COVID-19 infection includes the Phase 1 clinical trial in the U.S. for the aerosol dosage form. The main objective of the study is to evaluate the pharmacokinetics and safety of SNS-812 in humans. It is scheduled to enroll 44 healthy subjects and to complete the clinical trial in the first quarter of next year. Then, Phase 2 clinical trial can be started in the Q2 next year. In terms of the cooperation with the key laboratories in China, we are currently using the IND granted by the U.S. for our communication with NMPA, so we can follow China's zero-COVID policy to initiate the clinical trials in China. What are the opportunities for our SNS-812? The current treatment in COVID-19 includes Pfizer's small molecule oral drug Paxlovid. Although its market share in the U.S. market is as high as 90%, the recently released data from several important clinical trials was not ideal. In April, data on clinical studies of Paxlovid for the prevention of COVID-19 infection published by Pfizer showed that the drug did not work as expected, and no significant preventive effects was observed. In August, the prestigious New England Medical Journal NEJM published a clinical study covering 109,254 COVID-19 patients, which also showed that Paxlovid did not create significant therapeutic effect at treating patients aged under 65. In addition, although everyone has received vaccine, the COVID-19 prevention vaccine has decreased significantly over time due to the virus mutations. There is still a large and urgent need for effective therapeutic drugs around the world. Our SNS-812 has the advantages in COVID-19 treatment. Pfizer's Paxlovid is a viral protease inhibitor which block protease and inhibit downstream viral protein maturation. According to the past experience, for example, when treating AIDS or hepatitis C, viral mutations easily occur which leads to drug resistance. However, the mechanism of our SNS-812 is relatively unique, which starts from inhibition on the RNA of the virus. This will prevent the impact by viral mutations on efficacy. In addition, IC50 inhibition of different virus strains by Paxlovid is about 22 to 225 nanomolar, while SNS-812 is only about 0.05 to 0.5 nanomolar, which shows that SNS-812 has obvious advantages in efficacy. At the same time, we also know that Paxlovid has drug-drug interaction potential with 40 commonly used drugs, resulting in efficacy and safety concerns. The target of SNS-812 is relatively specific and can avoid similar problems. Next, I will share the reimbursement application progress of Fespixin and its post-marketing research. Following the expert meeting held by National Insurance Bureau, Oneness has completed the reply to meeting feedback in July this year. We have not received any further feedback from the Health Insurance Bureau as of now. We all know that Fespixin is the first-in-class innovative drug. CDE has completed the Health Technology Assessment Report. Fespixin is a breakthrough innovative drug with applicable reference pricing. In the comparison with the current standard treatment, Fespixin is more cost-effective. We are now waiting for further reply from the Health Insurance Bureau and will update to shareholders. We have collected doctors' feedback on post-market use of Fespixin in treatment of diabetic foot ulcers with 1,024 questionnaires received. The feedback showed that Fespixin is supported by good safety without drug-related adverse events, and it's easy to use. The average healing rate feedback by physicians is more than 60%. These are in alignment with our Phase 3 clinical trial results, which demonstrate the excellent efficacy of Fespixin in the treatment of diabetic foot ulcers. In terms of expanded use, although Phase 3 clinical trials did not include patients with dialysis, doctors now also use Fespixin in difficult cases of diabetic foot ulcer patients on dialysis, and the results have also been positive. At the same time, for some difficult and complex large area wounds, the doctor performs debridement and the use of Fespixin right after debridement, which also shows very good efficacy. This is a downgrading treatment strategy to promote healing. Let's take one clinical case for example. This is a 70-year-old woman who has been diabetic for 30 years with chronic kidney disease and hypertension. Her left knee has been amputated, and her right foot had a serious ulcer infection with an area of more than 60 square centimeters. The physician recommended a further amputation, but the patient was in tears hoping that the doctor could keep her foot so she could still walk. After the debridement, Fespixin was applied, and it was obvious that the wound gradually improved. And after 300 days of use, the entire wound completely healed. So the patient is very grateful to have Fespixin to help her wound heal. In terms of the cooperation plan of medicine, industry, academia, and research of Fespixin, we promote the special research of Fespixin and encourage doctors, obstetrics, academia, and research to participate in the academic and clinical research of Fespixin, hoping to further expand the new drug value of Fespixin. The current implementation of the cooperation plan includes 5 categories. The first is the lower limb venous ulcer, the second is the scar, the third is the biofilm of the wound, the fourth is the pressure sores, and the fifth is the research related to DFU. In our collaboration projects with medical centers and research institutes, we started with the exploration study in lower extremity venous ulcer, also called VLU. This study evaluates the safety and potential efficacy of Fespixin in the treatment of lower extremity venous ulcer. It is currently undergoing in Taipei Veterans General Hospital and Shuangho Hospital. For scar management, we have 2 projects, including clinical efficacy evaluation and the exploration and mechanism discussion. In the clinical evaluation, we explore the short-term and mid-term effects of Fespixin in improving scar after neck surgery or abdominal surgery. This study is undergoing in Tri-Service General Hospital. For the basic research, we use the translation research method to explore whether the other active ingredients of Fespixin can inhibit hypertrophic scar formation, and to explore the mechanism of scar microenvironment and the regulation of epithelial stem cells. This project is under collaboration with Taipei Medical University. The study on biofilms is conducted on a diabetic animal model to explore the efficacy of Fespixin in treatment of infection with biofilm. This study is under a collaboration with National Taiwan University Hospital. For pressure sores, we work with Taipei Municipal Wanfang Hospital and Kaohsiung Medical University to conduct an open-label study mainly to explore the efficacy and safety of Fespixin for wound healing in patients with vertebral pressure ulcers. We also initiated a project assessing the safety and efficacy of Fespixin for the treatment of chronic diabetic foot ulcers in patients with dialysis, which is performed at Cathay General Hospital. In addition, we hope to understand whether Fespixin can downgrade the foot ulcer severity from Texas Grade 3 on DFU patients. This study was conducted at China Medical University in an attempt to explore the effect of Fespixin in treatment of patients with ulcers classified Texas 3A and 3B. In the evaluation of new indications for radiation dermatitis, the post-market use of Fespixin has shown protective effects and reducing severity of radiation dermatitis. We established an animal model to explore the efficacy and mechanism of Fespixin. At the last investor conference, we reported the preliminary results on 3 animals. This quarter, we completed the study with 12 animals in each group. From this figure, it is clear that the sham control group had more severe dermatitis compared with the Fespixin or steroid groups. It is noteworthy that the group using Fespixin had a postponed dermatitis with less severity. Therefore, we can verify from in vivo studies that Fespixin can delay the occurrence of dermatitis and reduce its severity. We have also applied for a provisional patent in the United States. Once there is further evidence or the discovery of some further biomarker, we will file a patent application. We will prioritize to perform the exploratory studies in our subsequent plan to explore the clinical efficacy. We have also planned participation in an important conference for a continuous connection with healthcare experts and the international market. This year, at the 31st EADV Congress, we had a poster presentation. At the 51st ESD, our annual meeting, we also had a poster presentation. At the DFCon, we had an oral presentation. At the 30th Taiwan Society of Burn Injuries and Wound Healing Conference held in Tainan in September, we had an oral presentation. We planned the presentation for the Taiwan Society of Plastic Surgery and at the IDF Conference of the International Diabetes Federation, both of which are scheduled to be held in December this year. In July this year, we have completed the submission of a manuscript of ON101's pharmacoeconomic analysis. This manuscript is currently under review. In September, a review article on macrophage-regulating therapies to promote the healing of diabetic foot ulcers was also published on Pharmaceutics, a very well-known journal with an impact factor of 6.525. Also in September this year, we completed the submission of a manuscript on the analysis of post-marketing cases of ON101. The manuscript is currently under review. The post-hoc analysis of ON101 Phase 3 study is expected to be submitted to an international journal in the fourth quarter of this year. FB825's Taiwan exploratory trial data are also expected to be submitted to an international journal in the fourth quarter of this year. With a continuous publication planning, we hope to strengthen the research value and international exposure of Oneness new drug pipeline. At present, the licensing negotiations on ON101 are ongoing while the regulatory entry into global market is underway. Due to the confidentiality of the negotiation, no public disclosure can be made. Upon conclusion, we will make public announcement in accordance with the regulations. The above is our research and development report. I believe that everyone understands that it is very serious and rich, but we want to remind everyone about the investment risks. Oneness new drugs are global innovation. As high degree of innovation comes with high degree of risk, any R&D trial may create unexpected result. Oneness has tried our best to manage and respond to various risks, but we still remind investors the need to have a sense of risk. The deployment of the channel, the insurance payment, and the affirmation of efficacy after the launch of the new drug all take time, so please wait patiently. We believe that Oneness has the advantages of talent, capital, technology, and innovation momentum. We have abundant new drug research and development projects and continue to move forward. We still need the turning point before we enter the profit period. This turning point is the authorization reaching the progress, listed drugs entering the main markets, and continuously gaining drug authorization. The above is our report.
Thank you very much for participating in this investor conference. Due to the time limitation, today's online meeting is ending. For the questions raised by investors but not yet answered, we will provide response at Investor Q&A on our official website tomorrow before the opening of the stock market. Please refer to it. The next investor conference is scheduled in late December, and I wish you all have successful investment. Thank you.